It's so great to be here in person and see so many. So today I'm going to give you the talk, Clinical Neuroscience and the Heart-Brain Axis. So I became really interested in how signals from the body can shape how we think and how we feel when I was studying post-traumatic stress disorder in America. So I was based in Detroit and I was trying to see fear circuitry in the brains of individuals with PTSD. And it was during this time that the individuals would say the same thing. They say the world feels like an unsafe place. And it didn't matter if they were in a safe place, back with their families, away from trauma, or a dangerous place, that's how they felt. So my initial studies were manipulating the external context. I put people in dangerous rooms, I put people in safe rooms, and I was trying to look at the neuroscience of fear in the brain. And it was then that I really started to wonder about internal signals, because these individuals with PTSD had very different autonomic profiles. Sometimes their heart would race, they'd have hyperactivity of the sympathetic nervous system, they'd have a sweat response, they'd be very jumpy. And you'd see these autonomic signals that were sort of disregarded when we were trying to look at fear in the brain. So it began a fascination about internal signals, how they interact with brain dynamics, and how they shape our feelings, our memories, and other types of processing. So when we think about extraception, we think about senses such as vision, hearing, smell, taste, and touch, and these tell us something about the outside world. And in contrast, we have an internal sense, the sense of interoception. And there has been lots of debates about how to define interoception, and a white paper was written a few years ago that brought 100 interoception researchers together, and we came up with this shared definition, which is interoception refers to the process by which the nervous system senses, interprets, and integrates signals originating from within the body, providing a moment-by-moment mapping of the body's internal landscape across conscious and unconscious levels. And there are many different interoceptive signals, and the one that I particularly focus on is the heart. And my work tries to formally delineate different interoceptive dimensions so that we can see how these internal signals influence our thoughts and our feelings. So I systematically study these. I'm going to go through them in this talk one by one to give you examples of how we can study these in the lab and in the world. So first of all, there's the afferent signal itself. In this case, the one I study is the heart. So we can just look at cardiac signals in individuals. For example, we can take very basic measures such as heart rate and heart rate variability. And we can see that in different individuals with clinical diagnoses, you get these very systematic patterns of altered heart rate and heart rate variability. Heart rate, for example, is particularly high in individuals with schizophrenia. And this is a very robust finding. We saw it in our large patient cohorts, but it's been replicated many times before. There's also really interesting data to suggest that actually this elevated heart signal could even be premorbid, coming online before there are other signs of schizophrenia. In population studies where they've investigated individuals who are about to join the army, these are 18-year-old healthy men with no signs of schizophrenia so far. A high resting heart rate above 80% resulted in a 21% increase in the risk of schizophrenia when longitudinally tracked. And you also get these fascinating differences in heart rate variability. So the heart doesn't beat regularly, it actually has embedded within it these beautiful patterns of acceleration and deacceleration. And it's actually adaptive to have the variability in the heart signal. You want the heart signal to be reactive and adaptive. You don't want it to beat too regularly. And so you see this systematic pattern in individuals with different clinical diagnoses, where individuals with no clinical diagnosis has a high heart rate variability pattern, and you can see a systematic drop in all of these different clinical conditions, the lowest being emotionally unstable personality disorder and schizophrenia within our sample. So in addition to just tracking the afferent signal itself to see the different patterns, you can also look at the way it's represented in brain. And there's a number of ways that we've done this. We've looked at fMRI coupling with cardiac signal, but the example I want to give you today, which is brand new work that I've just started in collaboration with Mahinda Yogaraja, is looking at individuals with functional seizures. Now, we've heard of epileptic seizures, but functional seizures are fascinating because in behaviour, they look exactly like epileptic seizures. You can't differentiate it on behaviour alone. But when you look in the brain of individuals with epileptic seizure, you get these very classic patterns of focal activity in the brain, and you do not see these in functional seizures. So it really was a neural mystery about what could be driving these functional seizures. And we've been able to look at the cardiac signal represented in the brain to show it's a marker that predicts the onset of a functional seizure. So interictal means between seizures, and this heartbeat evoked potential measure is a neural measure where you can look at the amplitude of this neural signal that gives a measure of the cardiac processing of the brain of this cardiac signal. And you can see that between seizures, the interictal period, there's no difference in heartbeat evoked potential amplitude in either the epilepsy group or the functional seizure group. But the pre-ictal is just before a seizure onset, and you see this marked drop in this heartbeat evoked potential signal in the brain in the functional patients only. And it can really be seen as a measure of sort of body-brain integration, and you get this drop of this signal just before these functional seizures in the functional patients only. And it's when these functional seizures happen, they're often associated with feelings of dissociation. And it's sort of fascinating to think that one of the things that could drive dissociation and also these patterns of unusual behavior could be a degree of brain-body integration. In addition to the afferent signal and the neural representation of the afferent signal, we can also see how internal signals can actually change and shape the way that stimuli are processed. And this can be done in a number of ways, but one is to time lock stimuli to points in the cardiac cycle in my case, or the respiratory cycle in the cases of other bodily axes. And you can see, for example, when the heart is in active communication with the brain to see how this changes the way that stimuli are processed. So the brain actually is activated in time with each heartbeat, and this happens at T wave. For simplicity, I'm going to call this cardiac systole in the brain. And it's actually this point, which is a cardiac ejection point. You feel like your heart is beating, baroreceptors are activated, and the brain signals a degree of cardiovascular arousal. We can contrast that to R wave or late diastole, which is actually in between heartbeats. And the heart-brain channel is quiet at diastole, active at systole. And it's been known for many years that if you time stimuli to different points in the cardiac cycle, you can actually change the way that they're processed. And this was initially done in sensory stimuli, so you can give a pain stimulus at different points in the cardiac cycle, and you can actually see a reduction in the pain reflex response at cardiac systole when the heart-brain channel is active. So you actually get a dampening down of pain responses exactly when the heart is beating and the heart-brain channel is active. This early work was all confined to sensory processing. And I wondered if we could extend it into the domain of cognition. And this is in line with the theory of Lacey and Lacey, which are cardiac signals will have an inhibitory effect on stimulus processing. And I wondered if this could be extended into the domain of memory. So in my first study looking at this, I time-locked words to different points in the cardiac cycle, either systole or diastole, and I gave people a surprise memory test an hour later. So they didn't know that they needed to remember them, they just saw them, they didn't realize that they were time locked to different points in the cardiac cycle. And when we tested their memory for them via frequent recall an hour later, they were more likely to forget words which they encoded exactly when that heart was beating, consistent with the idea of an inhibitory or interfering effect of these cardiac signals. But I really wanted to see whether the cardiac effects would always be inhibitory in line with this Lacey and Lacey theory, or whether I could capitalize on the interest that I've got from working with people with PTSD, where these cardiac signals seem to be driving or influencing fear responses. And I wondered if there was something about the dynamic interaction between cardiovascular signals and brain that could also heighten or prioritize fear processing. So in a study where I took the same principle of time locking stimuli to different points in the cardiac cycle, this time I used fear phases and neutral phases, either at systole or diastole. And then I asked people to rate the intensity of these fear phases. And we were able to show that actually fear phases that were encoded exactly when the heart was beating at cardiac systole, heart-brain channel was active, then you get an increase in fear intensity ratings at this point. And this was in contrast to neutral phases, time-locked again to systole and diastole, where systole was associated with reduced intensity ratings for neutral items. There was a main effect of cardiac cycle in the brain, in the insula, but it was the amygdala that actually tracked the cardiac modulation of emotion. So this tracked the same changes of the heart on intensity ratings reflected in the brain and the amygdala, with heightened amygdala activity at cardiac systole for fear phases, and the reverse was shown for neutral phases. So we have the afferent signal, we have the afferent signal in brain, both of which can differ in individuals and relate to different types of behavior. And we have how the signals themselves can interfere with or facilitate different types of processing. But we have another interoceptive dimension, which is one of the ones that's maybe received the most attention, which is the accuracy or the precision with which these internal bodily signals can be detected and perceived. And these interoceptive accuracy dimensions have been looked at with tests historically focused on the cardiac axis, and tests have historically used something like the Chandri test, where people are counting how many heartbeats they have in a specified time frame. We record how many heartbeats they actually have, and then we look at ratio of how many heartbeats they report, how many heartbeats they actually had, and that serves as the accuracy score. This method's been highly criticized because higher knowledge of heart rates and sort of approximate time estimation can lead you to approximately the right conclusion. So this is seen as somewhat problematic. And the other method that's been used historically is one where you time stimuli either in sync or out of sync with the cardiac signals. And you see whether people can detect whether these tones are in sync or out of sync, and that synchronicity measure serves as a measure of interoceptive accuracy. That one is just really hard because it's an internal-external integration task, but there's no higher order knowledge that can help you do this accurately because the tones are the same rate as your heart. They're either exactly on the beat or just mildly time shifted off. So it's a hard task, and many people are at chance initially. But this task has been done in the scanner, and you can see that while many people find it hard, there is a degree of spread, with some people actually being very good at this and some people being very poor at this. And it's a fascinating, to me, individual difference measure, where some people are just very good at these interoceptive accuracy tasks and other people are not. And in the brain, you see that the anterior insula seems to be a particular hub for individual differences in interoceptive processing, where the volume of this area and also the activity in this area correlate with individual difference measures in this. And this has been known for a long time. It was Hugo's early seminal work. But to try and advance interoceptive accuracy measures, I've recently edited, along with Andre and Manos, whole edition of biological psychology devoted to interoceptive methods. So that's where the debates have sort of been historically, and there's lots of new exciting work on that now. But one of the things I find very interesting about these accuracy measures is, yes, some people are quite good at them, some people are not so good at them. But what seems to be fascinating is it doesn't seem to necessarily align with subjective report. That even these very hard tasks which can't be influenced by higher order measures. If you get people to rate their confidence on these tasks, then their confidence does not align with their actual ability. And in questionnaire measures as well, if you say how good you think you are or aware of you of these bodily sensations, they also don't align with these accuracy measures. And I wonder whether there is this greater divergence between accuracy and insight measures for these interoceptive dimensions relative to exteroceptive perception. So if the purpose of exteroceptive perception is to help you navigate the world, then as long as you're attending to something and as long as it's above a threshold, then most of the time you should know if you've seen something, heard something, felt something. Whereas our brains are being bombarded by internal signals all the time, and there seems to be this greater deviation between subjective insight measures and actual accuracy measures. And these divergences between these accuracy and higher order measures seem to be more exaggerated and larger in clinical populations. One of the groups I'm working with a lot at the moment are individuals on the autistic spectrum. And autism, as I'm sure you all know, is a neurodevelopmental condition where individuals display difficulties in understanding their own emotions and also the emotions of others. And also anxiety is really highly comorbid in this group. So estimates in the literature range from 23 to 60%. In the clinic where I was based in Brighton, I'd say it was 60% or more of adults in that service also had an anxiety disorder. So it's very comorbid and highly entwined. And you also see interesting neural changes in this group. For example, the insula has very different patterns of activation, and connectivity is also very reduced in the insula in individuals on the autistic spectrum. And the insula is this particular hub for interoceptive processing. So when trying to understand emotions in individuals with autism and how they understand their own emotions and the emotions of others, it has been said, and this is something I categorically disagree with, is that individuals who are autistic lack empathy. And I don't think that is true at all. The neural activity might not be quite the same in all circumstances. The self-report may at times differ, but as long as they understand, the empathy, in my experience, is very high. And when you look at the level of the body, you see this fascinating profile that's actually consistent with a higher empathy response. So when autistic individuals see other people in pain, then they actually have a higher bodily empathy response. They have more skin conductance when seeing other people in pain. And this effect was selective to pain images. It wasn't a generalized effect that happened to all images. And it made me wonder whether there could be an impairment or an alteration in interoceptive processing in these individuals, which may underlie some of the presentation of altered emotion processing in self and other. And if this is talk about autism, then I could go through all of these different pieces of evidence which align with the idea that there may be, in some individuals who are autistic, an interoceptive alteration or deficit. For example, intuitive eating, there's lots of cool work showing that knowing when you're hungry and knowing when you're full is very much an interoceptive process. It differs in different individuals, it maps onto BMI, and autistic individuals sometimes have different patterns of eating. Sometimes they forget to eat, they don't realize they're hungry, maybe they're not getting the hunger cues. Sometimes they overeat, they really like a piece of food and they just carry on eating it, and maybe they're not getting the same signals that they're full. Intuitive decision-making is some of my other work I'm not going to touch on today, where I show that intuitive decision-making can be influenced by internal signals, and autistic individuals sometimes make sort of very logical decisions that aren't necessarily as intuitive. And emotion processing and other processes, many of these can be linked to bodily state and interoception, and autism can have selective disturbances in a number of these different things which are potentially linked to interoception. So this was actually the first study way back in 2016 looking at interoception in an adult autistic group. So I hypothesized that autistic adults would have reduced interoceptive accuracy And I was also struck by what autistic individuals would say in the clinic, which is often they would say that they were overwhelmed by bodily sensations. So it wasn't that they didn't have them or couldn't perceive them. It was maybe that they were noisy and overwhelming. So I predicted a double dissociation between reduced accuracy and precision, but heightened self-report. And in terms of the accuracy, just using one of these straightforward cardiac tests, we were able to show that accuracy at the group level was impaired in these autistic individuals. You can see them represented in red, the matched control group in black, and you can see it wasn't every autistic individual, it was some. And this very much maps onto what we see in autistic individuals who have sensory differences. They're not necessarily uniform in all individuals. Some have them in some domains, some have them in other domains. And this data suggests that interoception may be a sensory domain where some autistic individuals really have a lack of precision, but some will not. But quite striking when we gave them a self-report measure, which just says how aware are you of interoceptive sensations, then you get this massive increase in reported awareness, which really contrasted against the reduced precision in individuals. And when I converted what I'll call an interoceptive trait prediction error, which was when I Z-scored both scores, and then I subtracted how aware or good people thought they were relative to their actual accuracy or precision to get an error term. This correlated with their degree of anxiety, trait anxiety. So if they thought they were very aware, but they had poor precision, then their anxiety was higher. So as a proof of principle initial study in healthy controls initially, I wondered whether we could train individuals to have better accuracy or better interoceptive precision, and whether this would in turn reduce anxiety symptomatology. So using 4 brief training sessions where we got individuals to do light exercise as a way to mildly increase their heart rate, so it was more observable and detectable. We then did the simple interception task with them. We got them to count their heartbeats in specified time frames, or we played them tones in sync and out of sync with their heart, and they had to judge whether they were in sync or not. But we gave them feedback on a trial by trial basis. You're correct, you're incorrect, you're correct. And also, because on half the trials they'd done this light exercise, they could also feel their heartbeat beating strong and fast, and this signal also returned down to baseline. But they were able to do these repeated training sessions as their heart returned back to baseline, and it was actually a means to stay within the interceptive channel with a fading signal. And we repeated this 8 times, but they also had a baseline, midpoint, and final assessments where we did not do any training manipulation, and we didn't give them any feedback. So we just wanted to see whether they had improved. And our main outcome of interest was anxiety, and whether this dropped in the individuals that underwent this interceptive training relative to a control group who were tested repeated times but did not receive any feedback. And here you can see that in terms of just counting their heartbeats in the interceptive training group, they were more accurate in this very simple tracking task. I do not find this result compelling at all, because maybe they were influenced by their higher order knowledge of heart rates. But what I do find more compelling is this in sync, out of sync task. It's incredibly hard. You can't do it correctly from higher order knowledge of heart rates. And there was a group difference that happened with increasing accuracy done from baseline to final only in the Interceptive training group. And we used a signal detection analysis as a means to look at D prime sensitivity, and this significantly increased in the Interception group only. But the measure that we're really interested in is looking at anxiety. And we found that trait anxiety dropped in our Interceptive training group, but did not in our control group. And then in terms of state anxiety, there was a drop, but it wasn't significant at the group level. However, the reduction in state anxiety was specifically coupled to the increase in interoceptive accuracy for both the tracking and discrimination task. So the more accurate they got with their training, then the more their anxiety drops. In addition, when we looked at the MAIA, which is a questionnaire measure to look at different aspects of bodily processing, those individuals who reported high levels of not worrying about interoceptive sensations and those with greater attention regulation to the body also showed significant drops in anxiety. And this tells me that there's three things which are important when it comes to interoception and anxiety. You need precision and accuracy into the signal. You need to not worry about the signal when you detect it, and you need to not overly focus on the bodily signal. Anxiety and depression are very much associated with a fixation and bias to the body, and it's actually adaptive to focus on the body when needed and focus on the world the rest of the time. So taking these principles forward, we ran a clinical trial. It was a registered clinical trial. It was randomized, and it had an active control group. And for our Interceptive training group, we ran fMRI in a subset of participants before and after the Interceptive training, and we looked at changes in anxiety. And we did this clinical trial specifically in individuals with autism this time. And we had a matched extraceptive training group where we taught the autistic individuals emotional processing. So they had sentences said in an emotional way, either a sad way or an angry way or a happy way, and they learned to identify what emotion was embedded in the way sentences were said. The idea being that lots of autistic individuals will say they get very, very anxious by not understanding or knowing how other people feels. So this was an extraceptive training that we could give them to help hopefully alleviate their anxiety through extraceptive emotion means compared to our Interceptive training group. And we also looked at anxiety change in this group. In terms of could we make these autistic individuals more interoceptively accurate, AD is our Interceptive training group. It stands for Aligning Dimensions of Interoceptive Experience. And blue is our control prosody group. And yes, we could make individuals who are autistic more interoceptively accurate. The D prime again is this very, very stringent internal external integration measure. So it was effective in terms of interoceptive training. But our primary outcome variable was anxiety. And we saw significant drops in anxiety in our AD Interceptive training group relative to our control group. And what was really striking, we just uncovered now and blinded the 1 year follow-up data, is that these differences were maintained at 1 year, which I was really happy about. And then according to a predefined criterion that we pre-registered ahead of time, which was our recovered threshold, which we defined as a clinically meaningful threshold of a 6-point drop in anxiety score, and also a score below 55. And when it's only a 20-point scale, 6% is quite a big drop. And according to this threshold, 30% of our Interceptive training group was said to recover, which for a hard to treat population of anxiety was a good outcome clinically. And then in terms of pre-post scanning in our Interceptive training group, this was done while they did interoceptive processing in the scanner, focusing on their heart. And we observed greater coupling with the right insula and this VMP-FC area. And this increase in connectivity between the right insula and this VMP-FC area was specifically correlated with the increases in interoceptive accuracy. We also saw increases in left insular connectivity between the left insula and this interesting ACC region. The VMP-FC region is very much implicated in control and autonomic control. I find this ACC region fascinating. It's also linked to autonomic signal. So when you regress an autonomic signal in the brain, then you see this ACC region also appear. So my interpretation of what Interception training is doing is it's helping with autonomic control via the VMPFC and potentially aligning autonomic signals together in the body, making them more coherent between the insula and this ACC region, and both of these reach correction. And then just hearing how the autistic individuals responded to this interoceptive training, I also find illuminating. So one individual hinted at a reduction in external sensory overload. So they said this beautiful quote: as the inner channel gets clearer, the outer channel gets more quiet. And autistic individuals, some of them feel very, very overwhelmed by senses in the external environment. They feel overwhelmed by bright lights, they feel overwhelmed by loud noise. And this suggests that actually increasing the clarity of the interoceptive channel may help dull the sensory overload. And it makes me think that the sensory processing is very much like a seesaw, and maybe it got a bit out of balance with a lack of precision to internal signals and the external ones dominated. And in this individual, at least, they seem to find that the clarity of interoceptive processing helped dull the overwhelming nature of exteroceptive signals. Another individual said, when I notice the impacts of anxiety on my body, I'm more aware of them and I'm able to reassure myself that it's just a physical reaction. I'm better at taking deep breaths and trying to slow my breathing and heart rate down rather than letting it spiral. And I think this speaks to first the not worrying part. So yes, you need precision into these signals, but you also need to not worry about the signals. And then this also, I think, gives insight into how it's operating. The early access into these signals allows you to regulate them. And thus, when your heart is just starting to ramp up and you're just starting to feel anxious, you can regulate them more easily than if you're suddenly only aware of these signals when you're already in the state of a panic attack or meltdown. And then the final individual said, I believe it's increased my awareness of hunger, and as a result, I remember to eat, drink, and go to the toilet, which is amazing because we hadn't said to them anything about extrapolation and generalization to other bodily axes. And it does suggest maybe that helping in one axis might have implications for another bodily axis as well. So does this have implications for new treatments? I focused on the heart, I focused on one behavioural intervention dimension, but I've got an article out right now in TICS along with Camilla Nord at the CBU in Cambridge, where we're talking about all different interoceptive dimensions in the body and how they may underscore different mental health conditions and how different actions of different therapies may potentially work through these bodily axes. And I do think it's exciting about thinking how different treatments that target the body pharmacologically, behaviorally, or other ways may be able to make differences in mental health treatments, and will these open new doors? And just to support that, I'm going to show incidental work done by a group at UCL, where they looked at the medical records of over 100,000 people in Sweden, and that they found that just doing an analysis without necessarily a hypothesis, but to find strong associations, they found that for severe mental illnesses such as schizophrenia, bipolar, and others, that taking medications such as statins that acted on cardiovascular function seemed to be protective against schizophrenia and bipolar. And these are conditions where you do see these altered cardiovascular patterns. So yeah, it's exciting for the future to understand more about the mechanisms and see what happens. So to conclude, interoception can be delineated across different levels and axes. Some of these levels correspond and others are dissociated, and dissociations and interoceptive alterations may be particularly prevalent in clinical conditions. I really think we need a normative understanding of all of these different levels across different bodily axes so that we can really see how they're altered in different clinical conditions. And hopefully this will give us novel insights into new treatments. So thanks to my lab, thank you to my old lab and the research councils, and thank you all for listening.